going on for many years, and have a medicinal chemistry background. So the program I'm going to talk today about is Lupinear Therapeutics, and specifically the small molecule portion of that is where I will focus. But I will touch briefly on the biologics, which is a new program as well. So the program's vision is to combine the strengths of NIH and industry together. And we do this by having investigator-initiated ideas for novel drug targets and having strong disease assays and models in place, and combining that information with advisors with extensive pharma experience that we've contracted NIH, as well as access to industry standard contract services, as well as access to NIH staff. And we build a team around you and by doing that. So the goals are to de-risk potential therapeutics to the point that industry will invest in them, allowing potential new drugs to reach patients as efficiently as possible. We want to identify the best ideas for translations in the NIH research community through this funding opportunity and the associated infrastructure. We do this by providing a combination of grant funding, non-dilutive funding, and as necessary resources, in terms of contracts consultants. They are typically lacking in most of our research community, small businesses, academic concerns, the full cadre. They're just not there, and they need to be added to the program. But the overall goal and the way we structure everything is to preserve your PI, mainly your institution who actually holds the grants, intellectual property to facilitate licensing down the road and make the handoff to industry or venture or whoever's investing to take it further down the clinic as smooth as possible. So as I mentioned, I'm talking today about the small molecule drug discovery and development. As the program is directed towards academics and small businesses mainly. The grant funding is provided to get you in all cases to the clinic. So we'll talk about where you can come in. You either come in discovery versus development phase, but in all cases, the end goal is to get all the way to phase 1. All right. So no matter where you're coming in, you will map out a plan from start to phase 1. The stage of development, I describe discovery phases. I'm still doing medicinal chemistry, or I'm still characterizing my final candidate compound, versus development stage is I'm really ready to start GLP tox today. And I'll be honest, I rarely find anybody who's ready to start GLP tox today. When they put the grant in, they almost always need additional drug dose range finding tox studies, or they need additional characterization of the hits to do the in vitro metabolism required now by the FDA. They need additional things. And we'll talk about all projects starting at a certain preparatory phase and how to do this. The whole purpose there, as I said, I'll talk about multiple times, gaining access to the NIH-funded consultants, contract resource organizations, and our expert project managers, science officers, and NIH staff is really what this program is all about. So if you have all that expertise and you're just looking to fund your own internal expertise, this probably is not the best program for you. But if you need this, the drug discovery expertise in any of the areas, then this is probably a good program for you. We do customize the program to you. This is our layout where we have each program has a lead development team that consists of the principal investigator, the consultants, and NIH staff. The one requirement that we have, because we have so many different disease areas, we can't provide that through contract services. You must have, as part of the grant, all of the disease biology, the assays, to drive SAR, the efficacy models, the disease biology expertise, all needs to be in the grant, either through your own institution, through collaborators, through contracts you have on your own. The sort of disease agnostic pieces that you may not have, we can provide. So we have contracts in place from everything from a relational database. So if you're doing SAR, we can put the compounds in and have the biological data in a relational database so that it's easy to access down the road, and both from following it point of view, but eventually for licensing point of view. We have medicinal chemistry. That our medicinal chemistry contract has 3 components in it. It has synthesis, it has in vitro ADME, and it has computational chemistry. Most people utilize all of those. Some people, depending if you may be a development project like, you may only need to do the in vitro ADME characterization of your final compound in the discovery phase, and that may be the end of it. So we can talk about customizing this to your initial project as needed. And I recommend everyone who is interested, after this webinar, you can certainly reach out to me and we can set up appointments to discuss your project in detail. We have contracts for Pk and toxicology, and the toxicology, as I mentioned, is both GLP and non-GLP. So we do both dose ranging and GLP IND enabling tox for phase 1. We do Pk in rodents and mice and rats, but we also do dog and non-human primate. Drug manufacturing formulation, we have 2 vendors there, MRI Global and Curia. They do synthesis, so process research to enable the phase 1 synthesis to eventually be done GMP. And then the API, if we can formulate API in a capsule, fantastic. If we need to do some additional formulation work to enable phase 1, such as a solution, or even in some cases we've done spray bright dispersion work, we can do that as well. Eventually, we get to clinical trials, and we have our own clinical trial contracts through Duke and PPD. Presently, are our contractors for doing our single ascending doses in phase 1 to get the first in human completed. So you have to ask yourself the question, do you have the expertise in-house and the resources in-house to do these activities? And if so, to what extent? Again, I often talk to people about medicinal chemistry. You know, we're not looking to supplant your team. We're looking to complement the team. But I will say, you know, we expect to hit the ground running. So this is an area where many times it's better to utilize our contracts for multiple reasons. If, you know, you need to hire a postdoc or someone to do the synthesis after the grant is awarded, well, the clock is ticking on your thing. And of course, if you miss the milestone and it stops, you end up having to get rid of that person. And that's not a good situation either. But we can complement it. So if you have existing people, they can help and be part of the LDT while still maintaining the synthesis effort at Curia. So again, talk to us about it on your particular set of circumstances, and we discuss what we think is sort of the best way to go forward. The program is trans-NIH in its nature, which means we have many different ICs. There are 9 listed here that have signed on to the FOA, which means they will potentially take projects that are in their mission. And we'll talk a bit about not every project can come into under every IC. So for instance, NIDA and NIMH really don't want to take development projects. They'll take discovery projects through BPN, but not development projects. And the rules for like SBIR are by each IC. So we have to, you know, so the rules I'll talk about today, you'll see in my later slide, are directed towards NINDS's rules. But if you're coming in as an SBIR under for indication, for instance, for NIDA, we should talk about what those are for the grant and what they have. Each of these ICs has information in the FOAs about what they're interested in and what kind of restrictions they have put on it. As I said, you can come in anywhere along the pipeline. Our exits are all geared towards either funding or external partnership, other grants. Things can end due to milestones. This is a milestone-driven program, and projects do end before we reach the phase 1. But in all cases, we are trying to get to phase 1. But we have had several licensed projects that have been licensed and internalized by pharma, and they take it in before we get to phase 1, and that's a win for us. It's a win for you that you have the partnership to take it all the way. All projects come in at what's called a preparatory phase. They start at in this two-phase mechanism, either at the UG3 phase if it's a non-SBIR, or a U44 if it's a SBIR program. We always form the LGT. We identify and conduct the necessary experiments and studies to meet the requirements for initiating MedChem or IND enabling studies. So if you came in at, I just have a screening hit and some assays, and you would be doing exploratory chemistry, really fleshing out whether the assays are working well, enough to drive SAR, showing me that the chemistry is working well. If you're coming in IND enabling, do we actually have the full data package to warrant the GLP tox studies, or are there additional go, no-go things we need to establish first? So we're going to establish those plans, all the stuff before the next phase of the award. And the progression will be based on administrative review. Each project can receive only one UG3 or U44 phase. Typically, in the execution phase, we can support up to 2 additional years of medicinal chemistry. So that means 3 years total medicinal chemistry if you were starting at the very beginning. I have essentially early stage hits. And the last 2 years of the program would be directed toward development at a minimum. Of course, that also means if you're coming in later, that we don't expect a 5-year duration in most programs. It will be faster, but that is how it works. As I said, all projects enter the preparatory phase. Somewhere along that discovery to development pathway our entry criteria are spelled out in the FOAs, and we have guidelines on budgets. And as I mentioned, all ICs, not all ICs accept development projects, so be aware. I should mention at this point, the UG3, UH3 is all comers. So when I talk about small businesses, SBIR eligible small businesses, we encourage to go to U44, but you may be a small business that's not SBIR eligible because of your ownership structure or other reasons. You just might not be eligible. You can come to the UG3, UH3 in that scenario. We can take foreign applications. We can have foreign components in the UG3, UH3. Again, if you had foreign components, you wouldn't be able to do that under the U44 SBIR rules. So there are multiple reasons you might come here even if you were a small business. But the budget rules are quite different. So if you are coming under the UG3, UH3 and you are exceeding more than $500K in direct costs in any year of the grant, any year, you have to lay out all years, both the UG3 and the UH3. If you exceed 500K in any of the years, then you have to do what's called a ARA process. You have to ask permission, which means you'll have to give me a fleshed out budget with a justification, probably 8 weeks prior to the receipt date. So don't wait till the last minute if you're going to be close to that to talk to me. If you're coming into the SBIR rules, this is direct costs. These are total costs of 500K per year, a maximum of 700K across 2 years. The UG3 can only be 1 year. This can be up to 2 years because in certain cases, if it takes 5 years, you would have a 2-year phase 1 potentially and a 3-year phase 2, but phase 2 can only be 3 years. And the max you can spend across the phase 2 again is 1.5 million in total cost, no more than 3 million across those 3 years. So keep that in mind. Different budget rules, different requirements for entry into these. Questions about these, please talk to us about it. Our next receipt date is in August, followed by one next in the February 2023. Applications for foreign organizations I mentioned are okay, but there is a second level review, and reviewers will assess whether the projects present special opportunities for furthering research programs through the use of unusual talent, resources, populations, or environmental conditions that exist in other countries that are either not readily available in the U.S. or augment existing U.S. resources. And you need to address that in your application if you're coming in from a foreign, as a foreign organization. Alternatively, if you have a U.S. collaborator, you can come in under the U.S. collaborator as the institute of record and be a foreign component under that. And the rules are a little bit different for that. You still have to justify the foreign component, but it's not the same second level review that is a foreign organization coming in. I talked about milestones. We lay out milestones very clearly. We will put in project-specific milestones with your disease biology on top of these. But if you can see how we sort of lay out most projects. The exploratory phase is typically the first year of the grant, and it's all around, if we were coming at this phase, are your assays working? Do you have a screening funnel in place? Do you use the target product profile well fleshed out? And do we have medicinal chemistry? Can we demonstrate SAR? It's not just binary, either active or not active. We've shown some improvement in potency from where we started. We have a plan for multi-parameter optimization, so we usually benchmark the compounds, a compound or two at that stage for in vitro ADME as well as potentially in vivo PK if possible. The second year of the grant, if you're coming into hit to lead stage, it could be your first year, but the hit to lead phase is about showing me in vivo pharmacology in your animal models, showing me advancing the SAR, trying to focus in on probably a series of compounds for lead optimization. It doesn't have to have all the parameters completed. It has to show that you can optimize the parameters and they're not completely orthogonal. The pharmacology doesn't have to be by the route of administration. It could be, you have a oral formulation plan, but you're doing sub-Q in the hit to lead stage while you optimize the molecules for oral administration. Lead optimization is about getting to that final compound, making the selection, getting everything into the single compound, establishing your PK-PD relationship, so your pharmacokinetic pharmacodynamic relationship, and showing a therapeutic index by running a rodent dose range finding study. That's sort of our cutoff to say I have a reasonable compound for moving into development at that point. The development phase has a pre-development where we complete the dose range finding, establish our, between the rodent and the large animal, we'll have established what we need for doses for GLP tox. We're sure that we can actually figure out what salt form and tautomer we want to put into the tox so that we are testing the same thing that's going to hopefully go into humans. We have a tractable route. It's not, 27 steps with a 1% synthesis yield that we just can't produce the material. And this point is where we really make sure you have to have your clinical consultant involved, your clinical specialist on your side that helps shape that target product profile. So I, again, encourage you to talk to a clinician as early on. You'll talk to NIH staff about it as well throughout the program, about what really needs to be available for the clinics. This helps us make sure that we're not missing anything in toxicology. If there's a target tissue we need to pull out that's not part of the normal panel that we would do in GLP tox, we need to know that before we've run the tox, not after. And eventually, we'll do scale up, we'll do GMP synthesis, we'll make the clinical trial materials or have them ready. Typically, drug product is most times done at the clinical sites because phase 1 are very small numbers. It doesn't make a lot of sense to make a finalized drug product or even attempt to at this point. When you get to phase 2, it will make much more sense to do that. So that's what our milestones look like in this program. Again, the maximum duration of the grant is 5 years in combination of UG3, UH3, or U44, phase 1, phase 2. The difference is, that phase 1 contract could potentially be initiated at the end of year 5. And continue on through year 6, something after the grant ended, because the contract and the grant cycle don't necessarily have to be 100% aligned in that regard. But that's typically where we're at. We have often accomplished this within that 5-year time from start to finish. So as I mentioned, we try to make sure your intellectual property stays your intellectual property. So the inventorship is determined by U.S. patent law. There's no way around that. But Blueprint is not seeking a stake in your IP by the design of the program. So prior to grant award, we're going to make sure you have upfront IP agreements in place with potential inventors that are contracted by NIH, as well as the people you have listed on your grant. So if you have multiple institutions, we're going to want to see you have an agreement. Who's taking the lead on filing patents? Where's the IP being assigned to? Because we're going to assign it to the institution of record on the grant. And so if the other institutions is the one actually executing it, then we have to make sure those agreements are in place and how it's all going to be done. Royalties, you handle royalties whatever way you handle royalties. We don't tell you to do anything in that regard. And IP agreements should aim for unencumbered IP, consistent with our program. NIH staff are going to work as advisors, facilitators, but we're not going to tell you what compounds to make. Even though I'm a medicinal chemist, I will not listen on your LDT and say, you really should make this compound. That's what the consultants are for. That's what the Curia contracts for. There are plenty of really smart people out there to go, I can certainly say, are you considering these issues? And if so, how? And, that will usually foster enough internal discussion on your part to find answers and to develop the intellectual property you need to move your program forward. The program has a website, and you can see this particular website. I seem to have lost the website at the bottom here. I apologize. Oh, so it's on the top. That's why. So I'm being blocked by my own thing. So the website's up here at the top, and there's a host of information on here, and I get a lot of questions about it. And I point you to this website because it not only has the Blueprint Small Molecules, but if you were to click up here, you could get to the Blueprint Biologics, or down here, you can get to Blueprint Biologics or the Device Blueprint program as well. But you could see what we have for projects. You can see what the open funding opportunities are. It goes over a lot of what we talked about, but it also has information about who is our external oversight committee, who are our consultants, who are our contractors, and all of that. So the Ignite program is a program I at least want to touch on for a second. Is if you don't have the preliminary data to enter our program, the Ignite program is designed to get you that preliminary data. So you may say, oh, I'm too early for this program. I don't have the leads yet. Well, there's an assay development and screening way here. So you develop the assay, run the screen, identify good solid hits. Or there may be, you need to do additional medchem to develop an in vivo tool compound to run your in vivo efficacy study so that you have good preliminary data. Again, this PAR 21122 can get that data. Or you may need to develop an animal model that you don't have yet. And again, there's a PAR to do that. This has regular receipt dates. This is an NINDS program, so it's really focused on NINDS rather than other, but there are similar kind of programs in other ICs available. Rebecca Roof is your contact. Again, this is a milestone driven, but it's an R grant, so it's not cooperative agreement. You'll need to have a clear milestone to go from those two phases. It needs to be quantitative, and it's essential to move the program forward. I always want to leave people with some helpful tips to move things forward. One thing is read the funding opportunities really carefully, because it tells you how your project's going to be reviewed. It tells you what things you should include, and it tells you what things you should not include. And there are things you can do that will result in your application being withdrawn before I ever see it if you don't follow the rules. So it's important to really understand what each FOA and NIH is asking for. The other thing is discuss the program with us. Ask for a meeting with me, ask for a meeting with the Home Institute's disease program director to really understand how your project fits into the overall portfolio in that disease area, as well as how to do the program BPN. Talk to me about it. I would always start with me if you're interested in this program, because I can facilitate what you need to get for additional information, whether it be from Institute PD or whether it be contacting SBIR for SBIR specific issues. Stick to the page limits requirement. You don't try to use, micro font or change the line spacing, because that can result in your application being being withdrawn. Make sure you put forth a solid scientific premise that is supported by your preliminary data, and that preliminary data is rigorously done. And you need to address the rigor in the research strategy section. Was it blinded? Was it controlled? Was it properly powered? Both from the preliminary data as well as the data you're planning to generate, you need to address that so that the reviewers understand it. Address any obvious criticisms of the chemistry or biology, because failure to do so, reviewers will assume you didn't recognize it was a problem and that they will ding you for that because you haven't done anything to address it. So if your molecule has a clear structural flag, you should say, my molecule has a clear structural flag. That's going to be one of the focuses of the SAR, is to do SAR around this to get around that as quickly as possible. If you have an animal model that isn't in existence yet in your labs, you need to say, this is going to be one of our early milestones to show that we can actually do this. Clearly indicate what will be done as part of the grant and what will be expected by BPN contractors. It calls for the FOA, when it comes to reading the FOA carefully, it calls for you to put this in a table along with some other information. Make sure you include budgets for all years of the grant, both the first phase, UG3, and the second phase, UH3, or phase 1 and phase 2 of the SBIR U44. Every grant cycle, I get an application, it seems, that they don't have their out years and they have to hit them up. And sometimes the whole grant is written as if they don't have the out years, and then they have to quickly rewrite the whole grant to include the second part of it. And that usually is not something that can be done quickly. Do submit your application early, because I can sometimes catch this before it's too late. We talked about this, explicitly discussing your rigor. Complete all your registrations 6 to 8 weeks in advance. So I know there's been some changes in the ERA and the registrations. Make sure you get that done and you're sure everything is working well, because doing things at the last second, they can throw some really lengthy roadblocks that I have no way of removing for you. Because as I said, consider the application early. If you're applying to UG3, UH3 program and you go over 500K, again, 6 to 8 weeks before, I need to see complete budgets with justifications for why you want to do that. Talk to me early. Talk to your own business and tech transfer or BD group to make sure you have a plan for funding patents and licensing. You can't be part of the grant to do that. Some things not to do not plan this to be your sole funding for your lab, because it's milestone driven. It can end abruptly. We run hit a scientific dead end, and we'll say we can't continue. We'll need you to come back in after you've got additional data that, out of scope for what we can do. Do not under-resource your budget to avoid the 500K limit that I just talked about. I would rather you come in early and give me a justification than you under-resource it and not be able to do it and miss your milestones because of that. Do not plan on the BPN contracts, as I mentioned, to do disease biology or one-off in vitro experiments. So if you come to me with a development project and you say, I just need to do an in vitro HERG, not a good use of our contracts. Our contracts cost a lot of money to put in place. If you're doing a programmatic thing, I'm doing, the whole gambit of in vitro experimentation on an analog or two, that's something we could potentially do. But if it's just a few one-off experiments, budget that as part of your grant. Do not code your application as a clinical trial if you plan to use the BPN contractors to do the clinical trial. This is always confusing to people, but even though you plan to do a clinical trial, if the clinical trial you plan to do is through BPN contracts, that clinical trial aspect is handled when we do the contracting portion of it. So the same forms you would fill out for a contract, for your grant, the contractor has to fill out when they're doing the actual contract proposal for us. So you can just say that. Now, of course, if you have human subjects for some other reason, you collect blood from students and you're utilizing it, then you have human subjects and you have to explain all the rules for how that's handled. But be careful on this. Again, this trips a lot of people up. I did want to touch base on the BPN Biologics program. As I mentioned, I'm talking about small molecules. That's been the ongoing program for the last decade, but this past year, starting FY22, we've launched BPN Biologics, a very similar program designed to do the exact same things and have the exact same receipt dates as the small molecules. There are different PIR numbers here. Mario Skiadopoulos is our program director for this and can certainly talk to you about this. So peptides, we say anything greater than 6 amino acids, you should think about the BPN Biologics program rather than the BPN Small Molecules. Certainly antibodies and peptides and proteins and gene therapies and cell therapies are very diverse. So the contracts we're putting in place are designed to have that kind of flexibility to do that. But the PK and Tox are all very similar to what we're doing. And sometimes we utilize the same contracts we use for small molecules. Likewise, the clinical trials may or may not utilize ours. And some clinical trials, it may not be appropriate to use our contract because you may need to target a certain patient population that only exists in certain places. If you're doing pain, there is a pain. Oops, let me go back up 1 second. Sorry. There is a pain program called the HEAL, Helping to End Addiction Long-Term program that has a suite of funding opportunities associated with it. The one that's very similar to BPN and was designed after BPN is the non-addictive analgesic therapeutic development program for both small molecules and biologics. It's a RFA NS 21-010, and the next receipt date is in June. There's a webinar for that program on April 28th, hosted by Marianne Palimonter, and you can certainly join that, and Wakanda can give you the link if you're interested in that to register. It is set up, like I said, just like it. The biggest difference is that we'll ask you about a target engagement biomarker in that program, and can you develop one as part of the program. So I'm going to stop now for questions and discussion. Those questions can be about the application process, around the review process, proposals and budgets, contracts, intellectual property. At the end, of course, you can always ask questions via email if there's not time to ask. And as sort of a separate aside, in case you're into gene therapy in there, and you know people are, our ultra rare gene therapy network, Urgent, is also having a webinar next week. And you can talk to them about that program. Chris Boshoff is your contact for that. So coming back to this, I'm open to questions now, and put your questions in the chat and go from there. Thank you all for your attention.
Okay, we have one question from Stephanie Coffin. Can you elaborate on the milestones? How frequently should we have them? How detailed? Do these all need to be outlined in the application, or are some planned during the preparatory phase?
So it's important to lay out a complete plan from wherever you're starting all the way to the clinic, if that's where you're going through our contract. So all the way through IND, enabling talks, and you say you do it. Part of laying out the table of who's doing what will dictate what you need to put in your application. So if you are saying you're doing stuff yourself, that should be well laid out in your application in there. If you're saying that you plan to utilize our contractors to do the work, you can just outline that in the research strategy section. You don't have to go into a ton of detail. The milestones will be established after, and I certainly can share the milestones that I laid out here for each step and what we typically have. You see them actually in the FOA for what the entry criteria for exploratory looks like. The milestones or generic milestones are there, but I have generic milestones for every step of the way. And I'm happy if you're interested in getting more. You don't have to have really extensive milestones laid out, but you should have a Gantt chart if you can. It's probably sort of one of the better ways to do it across time, saying this is where I'm going to be at each year of the grant, and here's where my go no go decision point will be at each one of those end of year time points. I think that's probably the best way to lay it out for the reviewers. We will establish milestones after the fact, and the reviewers are aware of that.
Okay, we're waiting for more questions.
Okay. I mean, if there are, like I said, I always encourage people to contact me and talk to me about their program. I'm happy to go through more um, extensively on one-on-ones as well. See, the question just popped up.
You got it, Chuck?
Yep, for a development stage API and candidate formulation for treating acute ischemic stroke, what translational efficacy studies does your group support? As I mentioned, disease biology we do not do as part of our program. You have to have that as part of your grant proposal, and you have to propose the ones that make sense. If you're unclear about what makes the most sense, I would say talk to NIH staff. This is where I would normally involve one of the science officers, someone like Jim Koenig, who I think does the ischemic stroke portfolio. I would probably involve him and say, what is the present day thoughts on this? I know a lot of models are looking for, in the stroke area, we're really looking to make sure that we see behavioral outcomes at a reasonable point after the event. And those are as important as seeing effects on tissue preservation. For hit compounds with some mechanism of action information but no direct target identified, would there be a mechanism for doing some target ID to assist the medchem development, such as in silico docking studies? So target identification is not part of the program per se, but if you had some putative targets, could we do as part of the medchem, the computational chemistry docking experiments? Yes, we could, and yes, we could try to do that. Phenotypic screening is permissible in the program. Obviously, it can be a bigger challenge to optimize molecules in a phenotypic way. But there have been successful programs within the BPN that have been phenotypic and moved on to candidates and licensing. Study section. Sorry, I should have talked about that. So our applications, both the BPN and BPN biologics, are reviewed by special emphasis panel of NINDS's review branch. So the BPN has historically always been a separate CEP and not part of any other review panel. BPN biologics, I did not sit on the review. It may be part of a standing study section under NINDS and then a CEP from that, but I'm not sure. You'd have to ask Mario for that, but certainly for small molecules is a separate CEP. So can I comment on how much preliminary data is needed for discovery phase of the project? This is always debatable, and you have to make a strong enough case that the premise is there. So you have good preliminary data that was done in a rigorous way. It doesn't have to be with your compound. So it could be you have great preliminary data with an antibody for this target, and now you have a small molecule that's interacting with this, and that's not, it's not a suitable tool compound to run in vivo on small molecules at this point. That's the point of the program, is to take it from where you are to getting tool compound to getting a drug. So you have to make that clear in your application that I have this data, whether it be from a literature compound and not your series, whether it be from other biological data um, out there, that here is why I believe this to be a viable um, target or biological process for which I want to intervene for the indication area that I'm describing. And you need to show, like, this is where the clinicians come in, that there's a high unmet need there, and that interacting in this way would be potentially adopted by the community afterwards. Yes, you can enter so can you enter the hit to lead stage with multiple scaffolds? Yes, you can. Um, the question is, if you're entering the hit to lead stage, that means you've sort of already showed me that you have up and running assays on day one. I have, you have your primary, secondary, counter screening assays, your cell-based assays, even your animal model stuff, all up and running, ready to go on day one. And you can show me metrics for that you have properly shown SAR for those scaffolds that demonstrates that I have a range of potencies. It's not binary, that I've improved the potency, that I don't have orthogonal SAR for in ADME, and they would say that the scaffold is really not viable. In the absence of that, you'd have multiple scaffolds in exploratory phase, where you'd say, how are we going to prioritize amongst these? We may benchmark them all for those kind of things. We may look at efficacy or activity. We may look at the ADME profile across multiple scaffolds and try to figure out which scaffolds to focus on. So I do say be careful on which stage you're coming in, but multiple scaffolds are allowed in both exploratory and hit to lead. In fact, we encourage you in the early stages to include as many of the different possibilities as possible, because that helps us to build models and everything else. Is there an interest in combination therapeutics? I think, generally speaking, there is an interest. However, pragmatically, it's difficult to execute in this program. And I would encourage anybody interested in combination or repurposing to talk to me in advance, because this may or may not be the appropriate program for your project. Um, we have limited resources, and a lot of time combinations require that you do each one individually first and then combine them second. How much characterization of compound metabolites can be done by the contractors? Will they isolate sufficient metabolite to test for potential activity? Isolate, no. We won't do that. We will sometimes synthesize putative metabolites to identify them in things. So typically metabolites, if you have a late stage project, meaning you're getting down to this is my compound, metabolite idea makes sense, right? You want to know that, and we do it routinely before we go to the dose range finding tox studies, to know what our metabolites are across multiple species, typically in microsomes or hepatocytes, and most times hepatocytes, because it has multiple pathways for metabolism. And we'll look at that and say, do we have any unique human metabolite and that's not covered by the species? Because obviously every metabolite we see in the human, we want to make sure we either cover it in rat or dog or both, preferably. If we're unsure, so when we do metabolite ID, they do MSMS analysis. So the first level is we just say, well, this is a hydroxylation somewhere in the molecule. This is a glucuronidation. This is that. They can analyze the MSMS data and try to do structural elucidation on the metabolites. Again, unless the metabolites are of sufficient quantity, meaning if it was a major metabolite, obviously we would have to a major metabolite as defined by the FDA. If it's a small metabolite, and it may or may not be relevant to do at this stage of the program, but we still may identify some just to know for sure what they are. If combination therapeutics are a no-go, would a bifunctional single molecule be of interest? Bifunctional single molecules would be fine, because again, you're only characterizing at the end of the day a drug, a single drug, with multiple, more complex pharmacology. The issue, like I said, the issue with combination therapeutics is typically it requires sort of two separate programs and then combine them into an actual drug at the end. And so again, we should talk about it if you want to know a particular thing. I can, with more information, I can speak to better. If there are no further questions, again, I want to thank everyone for coming today. I do really appreciate everyone's interest. Um, we can't be successful without you all and your great ideas. And I think the program is really beneficial to the community. So I've been doing it for over 10 years now, and we've had, a fair amount of success, including some projects that are now in phase 3. So I hope yours project will be the next one that's successful moving down. So please talk to us about it. Thanks to you, everyone. Enjoy the rest of your day.
And yes, this will be recorded and posted on the website, Marie. So you'll be able to see the slides there.
Yeah, well, I typically print out a PDF version of the slides to distribute to all the participants. I can do that immediately. The actual recording of this talk takes a while because we have to put all the subtitles in and make it ADA compliant. By the way, sorry. So we have to, before we post that up. So that takes a little bit longer.
But Rakonda should have all of your contact information, and we can send you the slides if you need.
Rakonda manages my calendar, and anybody that comes into me, the next thing I got to do is forward the request onto Rakonda. So if you're interested in having a talk with me, if you CC Rakonda right out of the gate, you'll circumvent one step in the process. All right, thanks all. Enjoy the rest of your day. Stay safe.